Hello, everyone. My name is Nate. I own the website MCAT, where I offer a whole bunch of free resources, tips, cheat sheets, videos, so on and so forth to help you in your studying of the MCAT. Today, we're going to be just giving a relatively brief, for my standards at least, summary of the postprandial state, where we're going to be covering aerobic respiration, anaerobic respiration, glycogenesis, and then fatty acid synthesis. We're not necessarily really going to be talking about what's going on within each of these processes, just the main goal as well as how they fit into the postprandial/fed state. Of course, as usual, I have a link in the description that is not only going to take you to my website, but also one for the notes and graphics that I'm going to use throughout these slides. So you can find that in the link in the description. Just to talk about what's really going on in the postprandial/fed state. This is when you've just eaten a meal, you've just eaten a glizzy, a hot dog, whatever hot dogs are made of, who knows, but that's one of my favorite foods on the planet. Shout out Costco. So once you've just eaten one of those hot dogs, you're going to have a high blood glucose level. You're basically, in a way, getting a free supply of glucose. So our body is going to prefer to use all of that glucose that we just received from that meal in order to start making ATP. We always need ATP. That ATP has to come from somewhere. Now that we've eaten a meal, that glucose from that meal is going to be what is turned into that ATP. So just in other words, all of the energy that we're making in the fed state is coming from the food in our diet, not our energy reserves. All of the ATP that we're making through aerobic respiration in the fed state is coming from hot dog glucose, in other words. So we're going to be running aerobic respiration to produce as much ATP as possible with all of that glucose. And then if we do have excess or too much glucose, then we're going to be storing the rest as glycogen or fatty acids. So basically in the fed state, we're going to start off with a whole bunch of glucose in our blood. Our body's going to release insulin, and that insulin is going to cause us to remove that glucose from the blood, either to turn it into ATP through aerobic respiration or storing it as glycogen or fatty acids in the liver. We're basically just using all of this glucose that's in our blood. So just to show you what this looks like within the cell, we're going to start off by throwing a whole bunch of glucose into the blood. All of this glucose from that hot dog that you just ate is now in your blood. And then our body is going to be releasing the hormone insulin in reaction to having a whole bunch of glucose in the blood. What this is first going to do is it's going to cause all of our cells pull that glucose into the cytosol using a glucose transporter called GLUT4. GLUT4 and insulin are going to work together to pull glucose into the cell. Once that glucose is inside the cell, we can start aerobic respiration. So we're going to turn that glucose into pyruvate via glycolysis. We can take those two pyruvate that we just produced, and in certain conditions when we have oxygen present and we can more so prioritize the efficiency of ATP production instead of the speed of ATP production. We're going to talk about that here in a second, just really quickly. We're going to ship that pyruvate into the mitochondrial matrix across those nasty membranes, where we're going to send it through the pyruvate dehydrogenase complex. Once we form this acetyl-CoA, we're going to combine it with a molecule called oxaloacetate to form citrate, which will thus allow us to send it through the citric acid cycle or the Krebs cycle. As a reminder, since glucose is a 6-carbon molecule that produces 2 pyruvate, and those 2 pyruvate are going to become 2 acetyl-CoA, we're essentially running the PDC and the Krebs cycle twice per glucose. So you'll just want to double all of those products in the Krebs cycle, since the Krebs cycle is happening twice, since we have 2 acetyl-CoA. Some of these processes will have produced some ATP that our cells can immediately start to use for whatever they need, but they will have produced a whole bunch of these NADH and FADH2 electron carriers. We can use a shuttle to ship the glycolytic NADH into the mitochondria, but there's also going to be a whole bunch of NADH and FADH2 produced in these mitochondrial processes as well. So now that we have a ton of these electron carriers, we can send them through the electron transport chain. The goal of the electron transport chain is to take these electrons, use the energy that these electrons carry, and pump a whole bunch of protons with that energy. This is going to create a high concentration of protons outside of the mitochondrial matrix in this region called the intermembrane space. And once we have a high concentration of protons in this intermembrane space, there's an enzyme called ATP synthase that is going to allow those protons to move down their concentration gradient into the matrix, which is a favorable movement. ATP synthase is going to couple that favorable movement to the unfavorable formation of ATP. So that's how the electron carriers and electron transport chain work together to help ATP synthase produce ATP. It's through this proton gradient that we're creating. However, if we don't have oxygen present or we need ATP at a super fast speed, instead of sending pyruvate into the mitochondrial matrix, we're going to instead run it through fermentation. In this scenario, all of these mitochondrial processes are no longer useful because we don't have oxygen, and oxygen is required for all of these aerobic mitochondrial processes. So as such, NADH, which is usually supposed to go to the electron transport chain, is now useless to us. So what we're going to do is we're basically going to recycle that NADH into NAD+, and NAD+, as you can see up here, is a reactant in glycolysis. So fermentation is basically our body's way of just saying, I'm not able to make a whole bunch of ATP through the mitochondrial aerobic respiration, but I still can make some with glycolysis. And fermentation is the way that we recycle all of those reactants that we need for glycolysis, NAD+. And there is also a way for us to ship this lactic acid to the liver and recycle it into glucose. That's a process called the Cori cycle. So basically, anaerobic respiration, which is glycolysis and fermentation, allows us to keep making ATP using glucose, even if we don't have oxygen present or we need ATP super quickly. Anaerobic respiration is super fast. It's just inefficient. We're only able to produce 2 ATP per glucose in anaerobic respiration compared to 32 ATP per glucose, roughly, in aerobic respiration. So now we might get to a point where we have been running aerobic respiration in all of our cells a ton, and we now have plenty of ATP. So in this scenario, whenever we have excess glucose still left over in the cells, the liver specifically is going to have a glucose transporter that is going to be shipping glucose into the cells of the liver in order to store this glucose as glycogen. GLUT2 is only going to be active when we have excess glucose, and it's going to help the body stimulate insulin to help keep removing insulin from the blood. So GLUT2 of the liver is going to help us really start kicking in glycogenesis. Glycogenesis requires the input of ATP to store glucose as glycogen, but that's perfectly okay because now we're at a point where we have plenty of ATP in the cell. This is largely happening in hepatocytes, but there are some other types of tissues that are capable of doing glycogenesis as well. Within the liver, we might also have excess acetyl-CoA, which we can decide to store as well. So what these liver cells are going to do is they're going to move this acetyl-CoA out of the mitochondrial matrix into the cytosol, where we can run them through fatty acid synthesis, which is going to use ATP to store this excess acetyl-CoA as a fatty acid. Specifically, within humans, we stop whenever we form the fatty acid called palmitic acid. So basically, we've laid out this entire process where in this fed state that we have a ton of glucose in the blood, we're going to be using that glucose to form ATP through aerobic respiration. And then when we have enough ATP, the liver is going to be storing that excess glucose as glycogen, and it's also going to be storing excess acetyl-CoA as fatty acids. Basically, we're removing glucose from the blood and doing a whole bunch of different stuff with it. One final portion of the fed state that's important to note is the pentose phosphate pathway, where insulin is also, in some cases, going to help us stimulate the production of NADPH or nucleotide precursors. NADPH specifically is going to be needed for fatty acid synthesis. It's also helpful in protecting the cell against reactive oxygen species. And then, of course, nucleotide precursors are going to be important for the synthesis phase of the cell cycle, where we're going to be making a complementary strand of DNA. So this is just another process that's promoted by insulin, the main fed state hormone, specifically in regards to metabolism that we're going to need for fatty acid synthesis. So now I'm going to show you all of that exact same formation, just in a slightly different way. On the right, we're going to be talking about all of our normal cells, your immune cells, your skin cells, your small intestine cells, so on and so forth. I do want to note really quickly that while we're talking about all of these cells, that red blood cells are one of those exceptions that the MCAT loves to ask about. Red blood cells, whenever we're talking through this, are only going to be doing anaerobic respiration because they do not have mitochondria. But basically, all of these other cells in the body, what they're going to be doing is they're going to be taking glucose from the blood and using GLUT4, they're going to ship glucose into their cytosol whenever we have high glucose conditions. High glucose conditions and low energy, low ATP levels. And once we have this glucose in the cell, we can send it through glycolysis, the pyruvate dehydrogenase complex, Krebs cycle, and then send all of those electron carriers through the electron transport chain, and finally allowing us to utilize ATP synthase. Just the normal parts of aerobic respiration is what all of these cells are going to be doing. And then, of course, there are going to be some scenarios that we can't send this pyruvate into the mitochondria. So what we're going to do is run it through fermentation, which is going to produce lactate, or in yeast, they produce ethanol instead of lactate. And this lactate is slightly toxic to humans. So we do have a way of actually shipping it to the liver, these hepatocytes. And once this lactate is in the liver, we can turn it back into glucose in a process called the Cori cycle. So basically, fermentation and anaerobic respiration allows us to keep running glycolysis, producing ATP in those conditions that we don't necessarily have oxygen, as an example. So that's what's going on in pretty much all of our cells, especially during the fed state. But the liver specifically, through the action of GLUT2, glucose transporter 2, can take excess glucose whenever we have high amounts of ATP. And it can take this excess glucose, ship it into its own cell, and run it through glycogenesis. Basically, we have plenty of glucose, we have plenty of ATP, we're going to start storing this glucose as glycogen. This does require the input of ATP, but like we talked about on the last slide, that's perfectly okay because now we're at a point where we have plenty of ATP. Glycogen is just the storage form of glucose. We can also take some of these glycolytic intermediates, and if the liver happens to need nucleotide precursors, specifically a molecule called ribose 5-phosphate or NADPH, it can also produce all of these molecules as well during this time. That's also a process that can pretty much be happening in those other cells at any time as well. So I'll make sure that's up on the right side as well. The liver might also have a bit of excess acetyl-CoA that it needs to do something with. And instead of just running it through the rest of aerobic respiration, the Krebs cycle and electron transport chain specifically, like our other cells would do, the liver can ship this acetyl-CoA into the cytosol, where it's going to be doing fatty acid synthesis in order to produce fatty acids. Of course, fatty acid synthesis requires the input of both ATP and NADPH as well. So the liver can store acetyl-CoA as fatty acids. It can also store excess glucose as glycogen. So the big thing that I want to highlight on this slide is notice that insulin is promoting all of these processes that push us away from glucose. It pushes glucose out of the blood to be turned into something, whether it's ATP if we're going down aerobic respiration, or we're storing it as glycogen or storing acetyl-CoA as fatty acids. Insulin's job is to remove glucose from the blood so that it gets utilized in some way. Glucagon is pretty much going to be doing the exact opposite. It's going to be pushing glucose into the blood. The last thing that I want to note while we're in this slide is that notice that fatty acid synthesis on the MCAT. It's also important to note that it can occur in adipose tissue, and glycogenesis as well we do see occur in myocytes. It's a fancy word for muscles. So hopefully that was a relatively quick summary of the postprandial or fed state. Of course, as always, you can check out the links in the description for all of the graphics that are in this video, as well as my other notes, my other videos, my tutoring access, my other free resources, tips, so on and so forth. So feel free to use any of my resources for whatever you need. That's all linked in the description.